Thanks, Tim, and good morning, everybody. I'm obviously James Graham, CEO and Managing Director of Recce Pharmaceuticals Limited. Today we have the usual corporate presentation that we're going to flow through, and I trust our friends at the ASX have done their very best to make it available for all market participants. There is new information in this presentation, so we'll just do our best to trust that the systems and processes in the background are reflecting that on the platform. But anyway, here we go. So by brief introduction of myself, I am really a representation by way of supporting good people and great science. The good people in this context, we briefly take a snapshot of our wonderful chairman, Dr. John Prendergast, who's chairman of New York Stock Exchange, a biotech company, managing director of a New York Stock Exchange or a NASDAQ a biotech company, about $500 million of value between them. And his doctorate is in the study of bacteria and blood diseases. So we've been working together, supporting this technology for a number of years, where he actually approached us saying, "Hey guys, I'm a former Aussie, how can we work together?" And that was a wonderful meeting between parties there. I started with the company as former head of, as the founding investor, supporting former head of research of Johnson & Johnson Australasia of about a decade in his retirement pursuit. His retirement pursuit was really trying to bridge that gap between the unmet medical need represented by the challenge of existing antibiotics, which work today and with repeated use no longer work tomorrow, and the unmet medical needs of particular indications such as sepsis, septicemia or blood poisoning, and multibacterial resistant infections. And we've made significant advances since that time. Chief Scientific Officer and co-founder of Recce itself is Michelle Delizia, we see there. And we have a wonderful team in the background of about 10 full-time persons. So what is Recce? So Recce, rather than in licensing technology, is built upon its own technology. Its intellectual property was beginning with the end in mind, developing and seeking to monopolize a new class of antibiotic. In fact, the first in over 30 years, some may even say 40 years. Our intellectual property has been granted by patents around the world out to about 2037. And our primary indication of sepsis, septicemia or blood poisoning, in fact, has the addition of market exclusivity supported by a QIDP designation awarded by the US FDA, which provides an additional 10 years of market exclusivity, or should I really say government-backed market exclusivity from time of potential approval along the way, and fast-track regulatory status all the way there. Actually, by law, it's under the GAIN Act. I know, a bit of a sidetrack. They have 30 days to review any data we submit to them and come back to us. No sending a data and a check and hoping to hear it, 12 months later. It's a 30-day legally designated window. And of course, we're very thankful for that. So we own our technology. Our technology is across multiple unmet medical needs, multiple methods of administration. We manufacture according to volume and quality suitable for all clinical trials. This is an indication of our pipeline. Our primary focus, as we see in further depth, is focused upon septicemia or blood poisoning. Basically, that's where bacteria gets in the blood from whatever it may be, a regular, general surgery or a basic accident. Any break in the skin, bacteria gets in and it spreads quickly. To my knowledge, we are the only clinical stage new class of antibiotic in the world. We are the only clinical stage new class of antibiotic for sepsis in the world. We are the only QIDP designated, as I mentioned before, in the world by the FDA for sepsis. And with that, we have significant advances, of course, through the phase 1 that we, as you'll see in moments' time, are now right at the end of. And with that technology, the opportunity to then, I wouldn't say pivot but to broaden. If we think of like a tree branch where, of course, it's, just one stick, how we then come out with multiple clinical phase sepsis or whatever it may be. You don't have to do the phase the phase 1 again when you're dosing via the same method of administration. So that phase that comes following is an exciting time for the company. In parallel, we obviously have a efficacy study using a different method of administration, and running in the background is advanced preclinical assets looking at multiple unmet medical needs, and we'll see what they may be along the way. So, sepsis. Sepsis is the most expensive condition treated in human health. Its costs effectively double that of the second most expensive. The patient population is large. We only go for large patient populations. We're not in niche, nano, non-economic areas. It's about 50 million persons per year. There's currently no drug therapies specifically approved for sepsis. For persons who do become a sepsis candidate, I've been a sepsis patient. I was, there was an accident. Bacteria gets in, every hour left untreated, the likelihood of survival of that person decreases by 8% each hour, and therefore the doctors must act quickly. So this is looking at the paradigm of challenge and what happens, or in fact, I'll just speak from a patient perspective, and I was the one that did survive. The statistics show it could have been quite against me. You present to the clinician, they see it showing indications of sepsis. This happens in every emergency ward, in any hospital all around the world. Doesn't designate or just discriminate, I should say, between healthy or unhealthy, young or old, male or female, or different ethnicities. It's prevalent. They do a pathology blood draw. They rush it off to the lab, and they're trying to do two things. One, work out what type of bacteria is causing your underlying infection, because any bacteria in the blood is bad bacteria. And what is that bacteria susceptible to, if anything, meaning of the remaining suite of antibiotics they can choose from? Is there a golden bullet to overcome that challenge? And increasingly, the answer is no. And that's the challenge. Bacteria have mutated. They always have, they always will. Antibiotics simply have not kept up. So Recce is designed as our suite of animal studies have supported to get in on patient presentation, to not worry whether it's a gram-positive, gram-negative, or a hypercellular mutated form, to immediately get on top of that infection at patient presentation. Let the diagnostics do what diagnostics do to tell you how bad, ugly, or how infected the patient may be. But don't let that infection run away, because every hour left untreated, the likelihood of survival is decreasing by 8%. So you've got to act quickly. So this is designed, as we see further in, as a new class, a class that is not reliant on traditional processes. All existing antibiotics are naturally derived. They're bacteria or fungi cultivated out. They only have mechanisms that are given to them. We've designed mechanisms with what we want: attracted to the protein of bacteria, bind to and interact with that protein, regardless of lipid outer layers, regardless of biofilms, regardless of gram stains, and regardless of their hypermutated cellular forms. And you can see in our presentations previously how well we work against the suite of escape pathogens, by example. And of course, economically, very attractive to manufacture. This is a independent study. We really, in this instance, look to it from a graphical perspective. Basically, as a small molecule, we permeabilize, or we enter the cell wall of the bacteria. We do a number of things once we enter. Well, firstly, obviously, we're attracted to, bind to, and then we enter. Firstly, we interrupt the cellular energetics. That means we interrupt, we stop irreversibly the beating heart or the lifeblood of the bacteria. And we do that faster than any other antibiotic known to date. Then, or relatedly at the same time, we stop the Z-ring complex, or basically the ability for the cells to divide and multiply. If you haven't got multiplying bacterial cells, you haven't got spreading infection. Above and beyond that, we obviously take advantage of the high metabolic processes that occur uniquely in bacteria. They don't occur in healthy human cells. So bacteria have always mutated to survive. They always will. But using these multiple mechanisms of action, it really gives potential and capability to overcome that hypercellularly mutating deadly bacteria that faces human health today. This is really expanding upon it from a discussionary perspective. We'll allow persons to read that in the background. So what happens when we are up against the healthy human cells? So this is really world first data completed by that group in San Diego. What we're really seeing here playing on screen is in the left-hand dish, we're seeing two things. One is E. coli, those little bacterial cells growing and multiplying, and those lovely, buoyant, healthy eukaryotic cells, the healthy cells that our compound seeks to avoid. Now, at 3,000 parts per milligram, which is quite a high concentration, on the right-hand dish there, what you're seeing is if you pick a bacteria and you watch it for a little bit as it plays very quickly, we'll work out a way to slow the presentation down, but you get the idea. Those healthy cells continue to multiply. They continue to grow in a completely uninhibited healthy state, whereas the bacteria in the same dish at the same time are actually not only not growing, they're disappearing, they're bursting. And those that do remain there, I'd say their ATP capability is entirely corrupted. And what we're really doing here is discerning between healthy cells. You don't want to harm them. Yeah, our intravascular studies have shown we don't. And here, the bacterial cells, which obviously is the ones that we have target for. So that's very exciting to see that the healthy cells remain uninhibited. This is looking at it from a different gram stain. So there were E. coli, here we're across to the opposite gram class of staph. These are a slower bacterial species to grow, so it's perhaps not as visually exciting, but in true contrast to the challenge that is human health. Staph, or the drug-resistant version MRSA or gangrene, is a major unmet medical need. You're basically seeing at the same time with a significant concentration of Recce, the healthy human cells are multiplying totally uninhibited, totally unimpacted, whereas the staph cells are not growing. In fact, I'd say they're dead. And that is an excellent thing for the case of getting on top of infections. So a nice little visual for us. Our human clinical trial, or should I say the human clinical trial done at Royal Adelaide CMAX to international regulatory standards, we announced we'd completed the 4,000 milligram intravascular dosing, and we have been dosing at 6,000 milligrams. That's 6 grams of compound infused in human blood or infused in healthy human subjects in a 1 hour period. You put 6 grams of sugar in your blood in a 1 hour period, you're going to know about it. So that's quite a lot of compound. This study is obviously the platform that allows us to then go into next phase studies. And as we come across to this next slide, because I can imagine there'll be members of the audience going, oh, why 6,000? I thought you were going to do whatever number they choose to pick. 6,000 was chosen because it is very high. As I mentioned, that is a lot of raw compound to have in persons. All patients treated to date have demonstrated good safety and tolerability of the compound, but really the study has been achieved. The objectives of what we sought to do, which is identify safety and tolerability, has been, the objective has been achieved. And what that really means is now we are well within broadly the efficacy range that we sought to achieve. The safety and tolerability that sets us up to the preparations of next phases, which phases include efficacy when intravascularly infused in human subjects, is very much underway. So this is a new slide. This is, as there was a number of previous new slides, quite market sensitive, but rather than me misspeak, I think we'll take it as read. Here is our second clinical trial happening in the background. This is a trial sponsored by West Australian Health Department. Multiple patients have been dosed in this study. It's led by world-leading clinicians, Dr. Ed Raby, and particularly Professor Fiona Wood, who was former Australian of the Year for her burn work, inventor of the spray-on skin technology. Cover your eyes, kids. This is quite a gruesome one. What we're really looking at there is, and many will come to this page and go, we want all the data. Well, we haven't got all the data for you guys. I'm sorry. What we instead here have is just a couple of examples, and we'll be feeding more examples out in the very near term. Some burn treatments, and burn treatments there we see on the left-hand pictures. horrible, terrible infected burn wounds. These burn wounds and the bacterial species underlying, without being specific to the picture, do in fact represent not only multiple species, but often these patients, in fact, in both instances, have had these bacterial species in their wound in one form or another for over 30 years. When we look across to after a short period of treatment, not only are we seeing a reduction in swelling, a reduction in red irritation, a visible reduction in bacterial colonization, and a healthy skin tone, and in fact, healthy closure starting to occur to the what was infected burn wounds. So I wanted to put them all up here today, but I thought it may be better to save that for a separate announcement itself, but we'll amuse those who think we don't have this type of stuff. We do, but when it's sponsored by the West Australian Health Department, it's sometimes done on West Australian government timeframes. So coming together, and we look forward to elucidating that. Supporting these advances, we have obviously an excellent suite of patents, our intellectual property, composition of matter and method of manufacture for family 1, plus our curative claims for bacterial species. Family 2 is preventative capabilities, plus methods of administration. So there we've seen obviously intravascularly administered in healthy human volunteers, as well as phase 2 topical efficacy, which is utilizing the same compound on top of the burn wound skin. It's also supported for nasal administration, inhalant, so on and so forth. And family 3 is our antiviral claims. So coronaviruses, Ross River viruses, influenza, HIV viruses, the list goes on, granted all around the world with market monopolies out to 2037. This is our manufacturing. We do this in Sydney's Macquarie Park. We have an excellent yield. We go from inexpensive, common, or not overly common, but inexpensive, plentiful materials, and we produce them we synthesize those in less than an hour to produce what we believe could be an extremely valuable high-end product. We have no expensive waste. Our capex to develop the facility, which is producing at both volume and quality to support phase 1 and phase 2 purposes, and frankly, the board among themselves saying probably phase 3 should at that stage come together, is really an achievement and an opportunity for Australian pharmaceuticals as a whole. Our capital position we're capitalized at about 156 million. We've got a good cash position. We obviously have R&D rebate opportunity, not only for our Australian activities, but we have that extended anywhere around the world. So we regularly do significant overseas studies, and those are supported to the tune of 43.5% by the Australian taxpayer. Our intravascular phase 1 study that we talked about there has been paid for, so the capital has been committed. It costs around $2 million. It's not expensive. The topical wound study cost us, that was great. We have no debt, caveat free. Our shareholders are obviously a mix of retail, sophisticated investors, and skin in the game by way of investment directors. So as an investment summary, we're an infectious disease company focused on the global health challenge of the infectious disease crisis, whether that be for antibacterial capabilities, hypercellular mutation, or superbugs, with indication and interest across the portfolio approach that we take in infectious disease areas such as antivirals and similar. We have a strong cash position. We have multiple near-term news flow opportunities, and an excellent technology supported by great people working to get ahead. And we really thank our shareholders and investment community for the time and interest in our company and stock, and we look forward to answering any questions that may follow. Thank you.
Thanks for that, James. We've had a couple of questions come through, so I'll just jump right in. Tony was asking, when do you anticipate results being reported on the preclinical Hpylori study?
Good question. So my hesitation there is one specifically. I'm not going to say tomorrow, the next day, or the next. But what I do know is when we say there was one study, we now, since we've moved into a clinical landscape, consider it as more of a suite of studies, or a program is a better terminology. So there's actually multiple studies that have taken place and have less and less materiality to our overall activity. So we will be reporting those, absolutely. We might be reporting other ones as well. But per study within a program that we seek to advance as part of our portfolio, we're transitioning away from singular study outcomes unless there's something catastrophic that would require a material disclosure, to more focus upon the program itself. And we certainly look forward to reporting that. I'd say in the very, very near term, but I can't be specific to today.
Right. No, that makes sense. And would that be the same case with the clinical study of R327 in sepsis patients, or is there a sort of a more material timeline that you have around there?
So now that we have achieved our objectives of the phase 1 intravascular dosing study, that positions us and I think we note actually in that slide of where we talk about why 6,000 milligrams, to step into efficacy type scenarios. I can't say whether we have or haven't dosed sepsis patients, but I can say that we have now a demonstrated platform of safety and tolerability when intravascularly infused in human subjects. So we are rapidly moving into phase 2 type activities, but as and when those would be disclosed, I'd say those will be disclosed pretty soon, but we'll see the specifics in due course.
Of course. So it sounds like investors are pretty curious about news flow. So maybe to cap it all off, can you give us, sort of 6 to 9 months of what investors can expect or what they can look forward to?
Gee, 6 to 9 months, and I simply think that because I was looking to where's the end of the year.
Anything can happen.
The end of the year is actually like within 4 months, and I was thinking, wow, there's going to be a lot that happens just in those 4 months. So what can we expect? Well, look, we've broadly completed our single 1 hour infusion intravascular administration. I am extremely confident that we'll, if we haven't already, been dosing in a next form being with efficacy type patient considerations within the calendar year. So yes, we would be starting, phase 2 type dosing within the calendar year. There will be further burn type announcements, or really an extrapolation of the burn wound study data. I think we had that on a slide there that said we'd expect to be reporting data sets and next chapters which are associated with that burn wound study. I would expect, all going well, there's likely a third, maybe fourth clinical study with an efficacy focus. I can't say the method of administration, but it would be a bacterial species focus, purely at unmet medical needs. That's all we're interested in. It's where price monopolies exist. And we don't play with generics in the space. They would be happening all within the calendar year. I don't think we'll be doing a capital raising. Got plenty of cash. I think there'll be an increased US focus. I happen to at this moment be in Australia for Australian clinical trial purposes, but we are very active in the United States in a variety of opportune areas. It really depends each and every, my CEO position, I look at all areas of the company and every one of them is very busy, but whether for this investment purpose, people take an interest or not is hard to say, but I do know we are getting ahead quickly and we're very pleased by the results to date.